Ladies and gentlemen, good morning from me, the Chair, Clive Page, and I'd like to welcome you all to this keynote lecture this morning. Unfortunately, Mene Pangelos can't be here in person. He's recorded his lecture, but he will be here at the end to take questions live, so please have your questions ready for later. It's my pleasure to introduce Mene Pangelos. And for those of you who don't know Mene, he's the Executive Vice President Biopharmaceuticals for Research and Development at AstraZeneca, and is responsible for biopharmaceutical R&D from discovery through to late-stage development. And his remit, it covers cardiovascular, renal, metabolism, respiratory, inflammation, autoimmune, microbiological science, and neuroscience. Prior to this, Mene served as the Executive Vice President of Astra's IMED Biotech Unit and Global Business Development. Mene joined Astra in 2010 and has since then led the transformation of R&D productivity through the development and implementation of an approach resulting in a greater than fourfold increase in success rates compared to industry averages. In parallel, Mene has championed an open approach to working with academic and other external partners, challenging the nature of academic industry collaboration, and has previously also held senior positions in R&D at Wyeth and GSK. Mene has been recognized in a number of ways. He holds honorary doctorates from Glasgow University, Imperial College London. He's a Fellow of the Academy of Medical Sciences, the Royal Society of Biology, and also of Clare Hall, University of Cambridge. He sits on the Council of the Medical Research Council here in the UK. He co-chairs the UK Life Sciences Council Expert Group on Innovation, Clinical Research and Data, and is a member of the Life Sciences Industrial Strategy Implementation Board. He's also on the boards of the Francis Crick Institute, the Judge Business School in Cambridge University, and Dizal Pharma. Mene was awarded the 2019 Prix Galien Medal in Greece for his scientific research and named Executive of the Year at the 2019 Scrip Awards in London. And most recently, Mene was awarded the honour of a knighthood from Her Majesty the Queen for his services to UK science. Mene also oversees the creation of AstraZeneca's new Global R&D Centre in Cambridge here in the UK, which is a state-of-the-art facility designed to stimulate collaboration, particularly between the UK life science industry and academia. So with that, I'd like to welcome Mene to give his talk today, which is entitled Translating Science into Medicines. If we can have the video, please.
Good morning. Good afternoon. I'm not sure exactly what time this presentation is going out, and I'm so sorry I can't be with you face to face, but I actually have to be in Cambridge for some business meetings that mean that I couldn't travel to Glasgow. My name's Mene Pangelos. I'm head of biopharmaceuticals R&D in AstraZeneca, and it's a pleasure to be able to talk to you today. And the topic of my presentation is about how we turn science into medicine. And what I want to do, just to get us into the groove, is just to remind you of how difficult what we do is and also how expensive it is. So if I ask the question, what's more expensive, developing a medicine or launching a rocket into space, in this case SpaceX, what would be the answer? Well, the cost of launching a rocket into space is around $400 million. So a pretty expensive endeavor. It's actually got a lot cheaper over the years. But the launch of developing a medicine continues to increase and is currently approximated at about $2 billion per molecule. So a hugely expensive endeavor, and you can see why it's important that when we spend dollars in R&D in AstraZeneca, we spend somewhere between $9 and $10 billion a year, we do it effectively and efficiently, and every improvement in our productivity has a big impact on our ability to generate medicines and ultimately generate a return on investment on the money that we spend. Now, why do I say this? The reason I say this is because when you look at the success rates, and I'll show you a graph from a paper that we published in 2014 in Nature Reviews Drug Discovery with a follow-on in 2018, you can see that as an industry, and if you focus on the very right-hand side of the graph, our success rates really are terrible. In AstraZeneca from 2005 to 2010, our success rate from first in human to launch was around 4%. So we were failing 96% of the time. That is terrible. Now, industry wasn't doing much better. Industry at that same period was somewhere in the region of 4 to 6% and has stayed pretty static actually out till today. So you can see that we're just not very good. And so what we wanted to do was this piece of work that was looking at every decision that we'd made from 2005, 2010, looking at data from around 250 different projects, what information we had, what types of decisions we were making, what questions we were asking in the labs, in the clinic, and as a consequence came up with 5 themes or 5 areas that we felt were the most important in terms of just trying to stack the deck a little bit more in your favor and improve your probability of turning that science into a medicine. And these 5 things are really very, very intuitive, but actually quite difficult to do on a day-to-day basis. So what are those 5 things? Well, the first one is right target. How well do you understand the pathophysiology of the disease you're trying to treat? Is it genetically validated? How good are the preclinical models? And are you asking yourself questions that are not just trying to prove your hypothesis, but also trying to disprove your hypothesis? Right tissue. When you have a molecule that it needs to inhibit a kinase in the heart or a receptor in the brain, do you know that your molecule is engaging with that target in the right tissue for the right amount of time, and also that it is, if it needs to inhibit or block a receptor, inhibiting or blocking it for the appropriate levels? So if you need to inhibit a kinase 80% in the liver, are you able to monitor that you're inhibiting 80% in the liver? If you're not, you never know whether you're testing a hypothesis. So being able to test and know that I'm testing my scientific hypothesis both preclinically and clinically through right tissue is incredibly important. Our programs don't move forwards unless they can demonstrate that through research and development. Right safety. Weeding out molecules that have got toxicity risks, weeding out targets and pathways that have got toxicity risks, and asking those difficult questions so that when you have a molecule that's not quite right, you just stop working on it and you move on to the next one. Right patient. Defining the patient population that is most likely to respond to your therapy. If it doesn't respond in that narrow population, it's not going to work. Your molecule is not going to work in a broader population. The best example of that is PARP inhibitors in BRCA ovarian and BRCA breast cancer. We needed to go into those select populations before we thought about going into broader or wider populations. And then finally, right commercial. And by right commercial, I don't mean is it going to be a $1 billion peak year revenues medicine. I mean, why would anyone want to reimburse it, pay for it? Why would anyone want to prescribe it? And why would anyone want to take it as a patient? So to do that, you've got to make sure you've got the appropriate standard of care. You want to make sure that you're asking the right questions, both preclinically and more importantly, clinically, so that when you get your results, hopefully when you get your phase 3 studies, they're relevant enough that actually regulators, payers, physicians and patients are all excited about the data and will want to get their hands on an innovative new medicine. By doing these 5 things, what the reviewer said in the first paper that we wrote was you're actually adding a 6R, which is right culture, which is changing the culture of the organization. And actually, if there was one message that I would focus on, it's actually focusing on quality versus volume. When we did this exercise, we cut about 50% of the pipeline in AstraZeneca when I joined in 2010, between 2010 and 2011. We focus on much less projects, but with a much higher quality. And what you can see is the culmination of that in terms of success rates from 2012, 2016, and 2016 and beyond. We're now up to success rates from first in human to launch around between 20 and 25%. So a very dramatic improvement from where we were at 4%. The industry has stayed pretty much where it is on average. And of course the challenge now is, even though we've got much better, we're still failing somewhere between 75 and 80% of the time. So there's still a lot of room for improvement. I don't want to get us to 50%, but actually if we get to 30%, that would be fantastic in terms of how many more of our molecules can become medicines. So what are the things that we're focusing on subsequent to the 5 Rs? The reasons why we're failing now are very different to the reasons why we were failing from 2005 to 2010. Invariably now, when we put a molecule into clinic, because we're testing right tissue, because we're weeding out all of the toxic molecules way before they go into the clinic. We're not failing because of safety. We're not failing because we're not testing the mechanism. We're failing because our scientific hypothesis is wrong. So we're getting good target engagement. We're blocking the kinase or the receptor wherever it needs to be blocked, but the hypothesis is wrong, and so the mechanism is not relevant in that particular disease process or that particular patient population. So enhancing our understanding of disease is incredibly important, and the more we can understand our disease, the more we can work on genetically validated or better validated targets, the higher the chances of us having successfully turning science into medicine. The second thing we need to do as a consequence of the first is actually making sure we have access to as many drug discovery platforms as possible, because many of the targets that we're coming up with now cannot be prosecuted easily with traditional small molecules or antibodies. The third thing we're doing is trying to come up with different ways of measuring endpoints in the clinic to improve our efficiency, our speed, the number of patients that we have to recruit into our studies. And the final thing is really transforming some of the ways that we conduct our clinical studies, and I'll show you a few examples of each of these. So let's start with enhancing our understanding of disease. This is incredibly important, probably the most important part of the talk, and probably the most important thing we do, because when you pick a new target to work on in research and development, you could be working on that program for the next decade, if not 2 decades. Normally we'll work on a program for somewhere between 5 and 10 years in research, and then maybe another 5 to 10 years in the clinic. So if you're working on the wrong target at the beginning, it could take you an awfully long time before you find out that it's the wrong target and it's actually irrelevant to the disease you're trying to treat. So it is a very important decision. So how are we doing that? Well, enhancing our understanding of disease can take advantage of all of the massive data sets we have, both internally within AstraZeneca, but also externally in the public domain, whether it's publications, whether it's patents, whether it's abstracts in scientific meetings. We're trying to put all of these data sets together and consolidate them in a way that our scientists can interrogate them and understand them. And we're doing that married with an investment in genomics, with our Center for Genomics Research that's taking large genomic data sets and actually identifying rare genetic variants through collapsing analysis that are driving phenotypes. We put all of these data sets together in something that we call knowledge graphs, which are an easier way for our scientists to manipulate these data sets and identify new nodes of interest or relevance for particular disease. And then from those lists of targets, we go into functional genomic assays where we do genome-wide screens using CRISPR gene editing to either knock out genes or upregulate genes that then give us an idea of which of these genes maybe have a functional consequence in relevant in vitro assays. We can then take the top targets from that into more complex in vitro screens. From there we go into in vivo, and from there we now have our targets that we're identifying that we think have better validation that run through our traditional drug discovery. We have around 40 new programs that we've started this way, 3 expansions of indications, and interestingly we've also published lots of really cool high-impact papers that I hope are accelerating the field and bringing new ideas to the disease areas that we work in. Now let me give you some examples. So this first one is through a collaboration with David Goldstein at Columbia University, where we're actually doing collapsing analyses of exomes from CKD patients, just over 3,000 CKD patients compared to 9,500 non-CKD controls. And this collapsing analysis was able to identify and enrich for genes that were known to be causative of CKD, genes like PKD1 and PKD2, but it also identified a series of genes that were not significant but were increasing risk of having CKD. And it was these particular genes that we were interested in, and we then put them through a series of in vitro experiments to try and identify whether any of these genes had functional consequences in vitro relevant kidney assays. And I'm showing you data from one particular assay here. This is looking at what happens when you transfect these particular genes into podocytes, and this particular gene that I can't name just yet, its name is actually just letters and numbers because that hasn't been published before. But this particular gene, when we transfected it into podocytes, into human podocytes, actually caused the detachment of the podocytes from the dish. So it was having a functional consequence to these cells that we thought was relevant. We've now subsequently actually knocked out this gene using an antisense oligonucleotide, and you can see the data on the right-hand side. This is in a diabetic kidney disease model in rats. And what you can see is that we're able to significantly reduce the amount of proteinuria in this particular animal model when we knock out that particular gene that was identified through this particular analysis. So really exciting data. This program is now moving through the research and development process and will hopefully be a candidate in the not too distant future. Let me talk to you about a second gene, and this is a gene that's very important in immunology, and it's targeting a receptor called the IL-33. It's actually targeting, a cytokine called IL-33 that works at the level of the epithelium, particularly important in the lung, and it's responsive to damage and irritants. And what we've discovered is that actually this cytokine signals not just through one receptor, which has been described before, called the ST2 pathway, which is the inflammatory pathway. If you block it, it's the anti-inflammatory pathway. But it also signals through a second pathway, through a receptor called the RAGE receptor. And we think that pathway is actually important in epithelial remodeling, epithelial repair, and mucus production. And so this mechanism is particularly attractive for us in asthma and COPD, where these two pathways actually both have relevance in the disease process. Now let me show you some preclinical, let me show you some more target validation data. So the left-hand panel just shows you IL-33 levels in bowel fluid from COPD patients compared to controls, and you can see that COPD patients have significantly higher levels of IL-33. And on the right-hand side, you can see a polymorphism that, when present, actually confers protection from early onset asthma. That's the blue line there. You can see there's a decrease in early onset asthma if you carry that particular polymorphism. So two additional bits of data that are giving us some reason to think that this could be an important pathway and an important cytokine in driving asthma and COPD. So here's some preclinical data. So top left is a model where we induce inflammation in the lungs of mice using an irritant, Alternaria extract. And what we can see, and we're measuring here IL-5, we've measured a number of other cytokines as well, but you can see that with increasing doses of a molecule called tozorakimab, which is an anti-IL-33 monoclonal antibody, we're able to reduce the levels of IL-5 produced by the lungs as measured in the bowel fluid. As I said, we've measured other cytokines as well, so showing the anti-inflammatory properties of this particular pathway and mechanism. But importantly, we've been able to show that this doesn't just have an effect on inflammation. The bottom left, we have in vitro cultures from COPD patients. These are cultures that have both goblet cells and basal cells. Basal cells are in purple, goblet cells are in yellow. And you can see that when we add in vitro the anti-IL-33 antibody to these cells, we're able to significantly reduce the amount of goblet cells and mucin that these cells are producing, and we're able to see a greater increase in the number of basal epithelial cells. All good things as far as a COPD patient would be concerned if this was happening in vivo. On the right-hand side, in a more complex culture, again derived from COPD patients, these are both video clips. You just can't see the cilia moving on the left-hand side because it's blocked by the mucus from the goblet cells, but you can see when we add the anti-IL-33 antibody again, we're able to clear the mucus and the cilia can now beat, and there is now flow through this organotypic culture. And finally, on the bottom right, this is again another assay using epithelial cells derived from human patients, either healthy volunteers or COPD patients. It's called a scratch assay. When you scratch a healthy epithelial cell from lung, you'll get the injury, but then it will narrow back down. When you do it on a COPD epithelial cell culture, what happens is the epithelial cells are not able to repair or recover, and so the injury stays wide. And you can see on the very right-hand side of that panel on the bottom right, when you have COPD epithelial cells in the presence of an anti-IL-33 antibody, the phenotype reverts to that of healthy tissue or healthy epithelial cells, and the injury is able to recover and become narrow again. So all data that suggest that this mechanism is important, potentially in asthma and COPD. What's exciting now is that there is clinical data that we have and other companies have also shown that it is relevant in both diseases in phase 2 studies, and so this is a molecule that we're very excited about, and is actually now in phase 3 clinical studies for COPD and in phase 2 studies for asthma. Let me talk a little bit about next generation therapeutics. So as I said, as we're coming up with completely unprecedented targets, often with targets that are not in the literature, where we have no idea what their function is, or even whether they're cell surface or whether they're intracellular, we have to have as broad as possible a therapeutic set of platforms that we can muster for our scientists. So we want to go beyond just traditional small molecules and monoclonal antibodies. And so you can see, I'm not going to go into all of them, but we have a variety of antibody platforms, whether it's antibody drug conjugates, Fab fragments, bispecifics, trispecifics. We have engineered proteins. We have programmable therapies such as antisense oligonucleotides, siRNAs, mRNA therapeutics, as well as in vivo expressed biologics, cell therapies, etc. Let me show you some specific examples. So the first one is an antisense oligonucleotide therapy. This is targeting a disease called TTR amyloidosis. There are two flavors of this. There's hereditary ATTR, which is extremely rare, and there's also wild type or sporadic ATTR. They both are caused by the abnormal folding of the TTR protein, which forms tetramers. It becomes misfolded, aggregates, creates fibrils, and these aggregated fibrils cause tissue damage, and particularly in the heart and peripheral tissues and organs. There are two diseases that are caused by this: ATTR-driven polyneuropathy and ATTR-driven cardiomyopathy. And they both have terrible prognosis, so actually therapies in this area are going to be very, very important, probably also severely underdiagnosed. Now, the therapy that we have, which is currently in late-stage clinical development, is an antisense oligonucleotide. This is a partnership with a company called Ionis, and it targets specifically the TTR gene, and it knocks down its expression in the liver very, very significantly. The polyneuropathy data, the 66-week data is just read out. We're able to see a significant reduction in TTR levels in the serum through 66 weeks. But more importantly, we're able to halt the progression of neuropathic impairment and improve patients' quality of life, which is unprecedented. And you can see that data on the right-hand side. This is something called the Norfolk QOL, Quality of Life Index, and you can see that people on standard of care were getting worse over time, whereas people on implantosome, the antisense oligonucleotide therapy, were not just staying stable, but were actually improving. If that continues out beyond 66 weeks, it's going to be incredibly exciting for patients, and it's going to be really a transformational therapy for this terrible disease. The cardiomyopathy study is currently still running and will read out in the next couple of years. Second program that I'm very excited about, which is in the preclinical stage, hopefully entering clinical development next year. This is actually creating human ventricular progenitor cells derived from embryonic stem cells, working with Ken Chen out of Karolinska and Harvard and Priscilla. Ken was able to define a way of getting human ventricular progenitor cells that are very high purity and very high efficiency. And we were able to take these cells and inject them into the hearts of animals that have had a myocardial infarction and show that we can see functional recovery in these animals through integration of these ventricular progenitor cells into the cardiac tissue. On the left-hand side, data from small rodents, so we can see an improvement in ejection fraction, how well these hearts are beating and pumping blood through the heart. In the middle panel, data from pigs following an MI, where you can see in pink these ventricular progenitor cells integrating into the pig myocardium at the site of injury and remaining there in functional in a functional way months out post-treatment, which is incredibly important. And finally, on the right-hand side, this is an in vitro experiment in slices from non-human primate heart tissue, where again we can see the human ventricular progenitor cells in the slice, and we can see them migrating to the site of injury through chemotaxis, which obviously is what we want to do if we're going to be putting these into the hearts of humans who have had or undergone a myocardial infarction. So as I said, really exciting data, really breaking new ground. We should be in the clinic with this program next year. The last program I want to talk to you about is a gene editing program, and this is targeting a disease called homozygous familial hypercholesterolemia. And this is a disease where mutations in the LDL receptor cause a dysfunctional LDL receptor, which means that people can't process lipids properly in their liver, and so they have very high LDL levels, and they have high risk of cardiovascular disease, heart attack or stroke. Now, unfortunately, this disease is driven by many thousands of different mutations, so you can't correct each and every one. So the approach we're taking here using CRISPR-Cas9 gene editing is that we're actually going to insert a corrected version of the LDL receptor into the human liver. Our proof of principle experiment is done in animals. I'm showing you that data on the right-hand side, where we knock out the mouse LDL receptor and we insert into the mouse the upstream of the mouse LDL receptor promoter, intron 1 and exon 1 of the LDL receptor. And then using CRISPR-Cas9, we insert the cassette that you can see there at the bottom left, which actually contains the corrected gene. And what you can see is a very efficient transfection of hepatocytes in the liver using this CRISPR-Cas9 gene editing process, such that we're getting about 20% of all hepatocytes being edited with the correct version of the LDL receptor. We still have to do a lot of steps to check that this is working well. We've got to get actually functional data on LDL function. We've got to get data on off-target correction. But overall, a very promising approach that we're hoping, again, will be entering the clinic in the next couple of years and potentially curing a disease which has got, again, a very poor prognosis. So, predicting better clinical success, the home stretch of the talk. So what are the types of things that we're doing? Well, we're using quantitative pharmacometrics and our quantitative modelers and statisticians to help us design better studies based on other people's data as well as our own experience with our own internal data. And as a consequence of the modeling they can do, in some instances they're able to reduce the size of our studies by half using modeling. They're able to reduce the number of doses that we tested in the clinic. And actually we're now seeing regulators actually accept some of these modeling data sets to actually mean that we don't have to run certain pediatric studies or drug interaction studies. So it can be a very powerful way of actually reducing the amount of work you have to do in the clinic and the cost. Continuous monitoring, another very interesting area where you can now use devices and sensors to measure things in real time in a continuous way. If you think about the way we used to measure blood glucose in the diabetes studies, patients would have to come in once a week and have their fasting blood glucose measured or their blood glucose measured. And now we can do that using continuous monitoring with a sensor where we're measuring people's blood glucose every 5 minutes continuously for 24 hours over a period of weeks. That means we can generate the same amount of data in weeks relative to what used to take us many, many months. So the speed of our studies, the size of our studies, again, very, very different, and again, much less burden on both patients and physicians. Novel endpoints. This is an example that we did in collaboration with GSK called COMPEX, which is an endpoint that you can use both in asthma studies and COPD studies. The traditional endpoint you would use in a study is an exacerbation, an event that happens when you have an asthma attack or an exacerbation in COPD. But they're quite rare and quite infrequent. So what we've done is collected a panel of indicators of what we think an exacerbation is, which mean that we can actually reduce the number of patients we need and the number of time that it takes to collect the complex endpoint, which makes, particularly in mid-stage development, much more efficient studies, and we think translates well to future phase 3 studies where you do have to use exacerbations as the endpoint. And finally, advanced imaging. And when you start to work on molecules and pathways that are involved in disease modification, you need to be able to have ways of demonstrating or proving that you are having an impact on disease pathophysiology. So this is an example of looking at small airway disease and COPD using CT imaging, but we can also look at mucus plugging, and we're looking at imaging in a variety of other diseases as well. But we're starting to use these advanced imaging endpoints to give us better clues and better data to support hypotheses that we're modifying disease processes versus just treating symptoms. Final thing I want to talk about is pioneering new approaches in the clinic, and this is something that I think has been accelerated by the pandemic. And the example I'm going to give you is a study called DAPR-MI, which is the world's first indication-seeking registry-based randomized controlled outcomes trial. And I know the regulators were very excited about this study, and we're excited about it because the way that it potentially transforms how you do a study relative to what would have been a traditionally very long, very expensive phase 3 program. So we're evaluating an SGLT2 inhibitor, Farxiga, in its ability to prevent heart failure or CV death following an acute MI. It's almost a 6,500 patient study, and traditionally you would do a study like that in around 25 countries around the world, and we're doing this study in just 2 countries. And the way we're able to do that in just 2 countries is because we're using pre-existing randomized registries that are part of a healthcare system's standard of care. The registries are called Sweetheart and MINAP. Now, what's great about using these registries is the patients are part of these registries and they're going to see their physicians as part of their normal routine care. And so we can incorporate their routine care into our study, or we can become part of their routine care without adding significant burden to the patient or the physician. And as you can see that it actually is projected to reduce patient burden by about 60% compared to a traditional heart failure study. We're also adding digital apps and sensors to enable us to measure things at home so the patient doesn't have to come on site. And we're also adding AI event adjudication, which means that we don't have to have external physician adjudication, but we have an algorithm that's actually helping us adjudicate. Obviously it has to be confirmed externally, but this again accelerates our ability to see events and to adjudicate events as we run through the study. The totality of this, that it actually reduced the cost significantly by almost 50%. And it actually significantly reduces the timelines as well. It says without impacting timelines, actually this becomes an easier study to recruit as well. So it becomes more efficient, more cost effective, better for patients, better for sites, better for physicians, and ultimately, hopefully, we still get a very well designed, very robust study. So I hope what I've done over the past 30 or so minutes is give you a sense of the types of things that we're doing to transform healthcare, to transform research and development, and ultimately to turn science into medicine. Thank you very much for your attention.
So thank you very much, Mene, for an excellent lecture, and I think we should be able to have Mene now live to take some questions from the audience. So thank you very much, Mene. Can you hear us okay?
I can hear you fine, yeah.
Good. Thank you for your really excellent talk. So I'm going to ask if there are any questions from the audience. Please, we have someone here at microphone 1.
Hello, Mene. Thank you very much for a beautiful talk. It looks like an exciting future for AstraZeneca and for pharmacology. My question is about understanding of disease. And I'm interested about your opinion on the amyloid hypothesis and whether or not amyloid is an appropriate target for Alzheimer's disease.
I think it's an appropriate target. I think the data from, it's not an area that we work in actively. I have a small neuroscience group, but you're going back to my history from 20 years ago. I think that from what I've seen from the recent data from Biogen and Eisai, the antibodies are doing something, at least in disease that's diagnosed early. So it is an appropriate target. Is it the only target? Obviously not. Is it progress? I think yes. And does it give us encouragement that other targets will hopefully have an impact on disease? And I think the answer to that is yes as well.
Thank you.
Okay, Amrita, and then I'll come to microphone 4.
Hi, Mene. Samrita Alawali. I'm the Secretary General of WCP 2023. I just want to say thank you from David and I for doing the recorded. We very much appreciate that you did that.
It's my pleasure.
And thank you for coming for questions. My question relates to one of the R's that you didn't talk about. You had the 5 R's at the beginning. You talked about most of them, but one of them was safety and getting the safety right. And at this Congress, we've had a few sessions focused on sex differences and also experimental design. And in fact, one of your colleagues, Natasha Karb, has worked with me we ran a workshop together. I just wondered what your thinking was about safety testing and improving the numbers of females in early testing, and then, of course, testing the drugs in women at later stages. Because we've got a big problem, haven't we? And understanding the toxicity in women is something that's been missing from drug development largely.
Yeah, I mean, I think, again, obviously you want to avoid going into women of childbearing potential because of the implications of that, particularly when you don't know as much about the safety of your medicine in humans, because at the end of the day, preclinical safety testing is exactly that. It's preclinical safety testing, and it doesn't always predict idiosyncratic toxicity. But when I talk about right safety, I talk about, in terms of our lessons learned, the easy stuff, avoiding safety signals that are actually inherently apparent in your programs, and we just kind of avoid them by lowering doses or doing something else, which then actually means you don't engage the target in the tissue for the right amount of time. And that was the legacy of our history, right? Is that you could get around your safety. What we do now is obviously we do a lot of structure-based toxicity investigation. We use big data, we use in vitro predictive models, organoids, we use human tissue wherever we can, and really try to weed out compounds that have an increased risk of safety issues before we get into the clinic. And what that's meant is that invariably I think our success rate going from, I haven't got the charts in front of me, but we're up at sort of 98% success rate in terms of getting into phase 2, which means we test our hypothesis. Now, ultimately, you've got to do the safety test. I'm not sure doing something in animals is going to help you with predicting safety in women, honestly. I think the best way of predicting it is by doing it in people carefully. But what you want to do is get rid of all of the idiosyncratic tox that you can avoid, so that then when you're putting things into people, you've got a molecule that really can be as safe as it can be when you're doing your clinical trials.
Gosh, I'm interested in your response. I mean, not testing, preclinical testing, at least you can include some females. There's not much of that done.
Fine. Of course you can include females, but I don't think it's going to protect you from the things that are coming up idiosyncratically. You'll get some level of protection. Of course, we have to do preclinical studies in female animals. We have to do it in pregnant animals as well.
Yeah.
But it's not. I don't think that those toxicity assays or toxicology assays where you're doing maximum tolerated doses are necessarily predictive of toxicity that you will see in the clinic.
No. Okay, thank you.
So microphone 4, please, and I'll come to 1 and then to you, Michael.
I'm Matthew Doog from New Zealand. A lovely overview, thank you. I'd like to share that with trainees and things as well. I'd just like to take you back to your first slide with those costs. I mean, your great efficiencies are going to have maybe a 50% reduction in costs, but how do we achieve the 10 or 100-fold cost reduction that we need for clinical care?
When you're talking about the cost, are you talking about the cost of going from first in human to launch?
No, I mean the end cost to the health system, which when often these medicines end up being used in relatively small populations, and the cost of that ticket on the spaceship is only affordable to a few.
But I think the point is that when you've got success rates that, when we're better than average, when if we're spending $10 billion a year on research and development, and we have a 20% success rate, it means $8 billion of our investment hasn't returned a medicine. in order to be able to spend $10 billion a year and generate innovation, you have to be able to recoup that with medicines that are priced fairly but appropriately. I can't tell you how we're going to fix the healthcare system and how we're going to make medicines more affordable. I can tell you that it's a very risky, very expensive endeavor. That was the intention of the slide. And therefore, while the medicines have exclusivity in terms of their patent term, they need to be priced at a price that's affordable, but also generating, a profit so that we can then reinvest in research and continue to invest in research and development.
Microphone 1.
Hi, Mene. Clifford Wolf, Harvard Medical School.
Hi, Clifford. How are you?
Great to see you. Your examples were all based on a single target, but there are many diseases where multiple targets may contribute, and a polypharmacology approach may be more appropriate. Is that something industry is adapting?
Yeah, so I mean, it's a great question, and our chemists, again, I think one of the reasons actually for not just working in chemistry and just in the chemistry and antibody space and working in the cell therapy space and the gene editing space and the oligonucleotide space is actually you can work on multiple targets. And actually, if you even think about the COVID vaccines, working on multiple variant vaccines, or something that is becoming more common and more prevalent for pathogens at least. Polypharmacy combinations, I think, is important. We're seeing the importance of it now, today, in cancer therapy, where you treat patients with multiple nodal points that are driven by their particular tumor pathway. In diseases where you can do that, obviously it's sensible. But what I would say is, you generally need to have an investigational medicine work on its own 2 feet first, and then you can think about combining. And I'll give you an example. In the cardiovascular and metabolic space, and dapagliflozin, which was originally developed for diabetes, is now a medicine that is had positive studies and approved in chronic kidney disease and in heart failure. That for us, which is an SGLT2 inhibitor, becomes a backbone to combine with other therapies and mechanisms. And so I do think that that's important. It's quite difficult to do polypharmacy. If you're a medicinal chemist and you're saying something, give me a molecule that hits 2 different receptors or 2 different pharmacologies, that's quite a challenging thing to do versus 2 individual molecules. But hitting multiple parts of pathways, if you can do it, very sensible, but challenging, I would say, Clifford.
Thank you.
Thank you. Michael.
Okay, Mike Spelling. So I've been associated with 14 drugs going to clinical development, and one, for example, DFMO for sleeping sickness. And the problem is that I'm also Secretary General for IUFAR, and there are 2 worlds for healthcare and 2 worlds for development and drugs. And what rare diseases are also, now the human population exceeds the number of nucleotides in the human genome. We're doing all sorts of rare diseases. And $2 billion is an impossible target for generics and other compounds. And so, as the Secretary General for IUFAR, I feel a responsibility in being able to see how we can develop faster drugs for rare diseases in partnership or not with pharma, because you've got all the expertise and you've also accumulated all the regulatory issues, which are now so complicated around the world. And I'm developing a generic for ALS, which is obviously a big issue. And is there a way in which pharma can also help and associate with certain academic developments in order to be able to get drugs for the developing world or for very specific rare diseases?
Well, so I think
sorry about that one.
Two very different topics there. Rare diseases is an area that I think is very interesting, and actually more and more pharmaceutical companies are going into the rare disease space. So rare genetically driven diseases where you can do antisense oligonucleotide gene editing, very low volume but high cost medicines. And I think you're going to see more and more of those because they're genetically validated in terms of the right target. They're great things to work on. They have relatively short development times and maybe a little bit lower risk, although again, endpoints, and there's lots of complexities. But I think what you're asking about actually is more developing world diseases, and low-cost medicines, and you know, it's very challenging, I would say. Again, I don't have a solution for that. I can tell you, having done the Chadox vaccine at not-for-profit, that was not an easy thing to do. And you know, would we do it again? I'm not sure. And actually, did it help being not-for-profit? I'm not sure it did either. I think there was a little bit sometimes a view that it was a cheap and cheerful vaccine for the developing world, and the mRNAs were the good, expensive vaccines for the developed world, which was absolutely not the case. So I think we need to be a little bit careful about the simplicity of. I think medicines need to be affordable and they need to be good value for money, but ultimately they also need to be profitable enough that they can allow people to reinvest in research and development.
Yeah, great. Okay, thank you very much. I've been in 3 pharma R&Ds. You've given a beautiful presentation. So
thank you.
Thank you. So time just for 2 more questions, please.
Thanks very much. Duncan Richards, Oxford. The range of modalities available now to prosecute targets is enormous, but a lot of the modern modalities are at a relatively early stage of development, which means the physical drug product that comes out at the end is often quite complex, and that can impact on you know, accessibility of that medicine. Can you comment at all about how you're thinking about the range of modalities that you think you can manage and make those accessible to patient populations in a meaningful way?
Yeah, I think like everything, it's an evolution. I mean, no one had heard about mRNA and viral-based vaccines up until 3 years ago, and now they're commonplace. And we've made them in volumes that we never dreamt of making them. So I think, like everything, when you have a medicine that works, you know, technology will evolve, manufacturing capabilities and capacities will evolve, and costs will evolve as well as scale of manufacturing improves. So first and foremost, the biggest challenge is getting something that works with these new technologies. So, you know, if you think now how many antisense oligonucleotide therapies have been approved, of which there's a number, right? It's becoming easier and easier to manufacture those, and the cost will continue to come down. It's the same for siRNA therapies. It's the same for mRNA therapies. Now, cell-based therapies, obviously right now, CAR T-based therapies, for example, are specific to an individual patient. They're very low volume, but you know, we're all working to try and generate off-the-shelf based cell therapies that will be much easier to manufacture and distribute and access. But I would say all of these you know, like every advanced technology, it's an evolution that takes many, many years. The exciting thing is that the tools that are available to our scientists now enable them to prosecute biologies and pathways and diseases that we wouldn't have been able to prosecute historically. And that opens up, obviously, a lot of new space and a lot of new opportunities.
Thank you.
So, final question, please, number 4.
Hi, Mene. Jeanette Watson from Jazz
Hi, Jeanette.
Pharmaceuticals.
How are you?
I'm fine, thank you. Lovely talk, thank you for that. You mentioned one of your R's was right tissue. So ultimately, that's human native tissue, target engagement, and showing function. So in the CNS area, that's a bit harder to do. So can you comment on how your CNS team achieved that R?
They don't. Well, so first of all, I haven't got a big CNS team, but they've just had a program that hasn't been able to achieve it, and they're not going forward. If you can't achieve it, we're not going forward. It's really very simple.
Okay. No.
Right? And I realize it's more difficult in the CNS, but
Yep.
I don't want to be spending lots of money on big phase 2s and phase 3s without knowing that I'm testing the scientific hypothesis. So I'm a little bit draconian when it comes to that. They have to develop PET ligands, they have to get surrogate markers, they have to have a way of demonstrating PK/PD relationships.
Okay, thank you.
It's really great. Sorry for being so blunt.
No,
I didn't expect anything less.
It's good that you've been blunt, I think, because I think it is a big challenge, obviously, with tissues that are less accessible, particularly for patients. So,
there are
Mene,
ways, the ways you do it is by looking for peripheral surrogates, that you think where you can kind of correlate the surrogate to brain in preclinical settings, and at least give you some confidence that the biology is mirroring in humans what you see in your animal models.
yeah. So Mene, again, just to reinforce what Amrita said, thank you very much. We're sorry you couldn't be here in person, but thank you very much for giving up your time this morning. I know you're very busy, to at least share the question and answer session with us. It's a really excellent talk and given us some food for thought. So thank you very much for your contribution, and we look forward to catching up in person soon. And if we can just have a round of applause for a really excellent lecture.